Hello, everyone, and welcome to today's live broadcast, Clinical and Economic Benefits of Faster Phenotypic Antimicrobial Susceptibility Testing, presented by Levi Kirwin. He is the Director of Commercial Marketing at Accelerate Diagnostics. I am Marjorie Torres of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Accelerate Diagnostics. For more information about our sponsor, visit accelerate-diagnostics.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located in the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left corner and follow the process to obtain your credits. Please join me in welcoming Levi Kirwin. I will now turn the presentation over to him.
So with that, again, thank you for coming. Thank you for being part of this today. I'm going to jump in now. And what we're going to talk about is really the clinical need for faster susceptibility results for patients in the ICU, for patients overall who are suffering from bacteremia. And we're going to look at some of the science, some of the recent publications that speak to the need for faster susceptibility results, how those empower clinicians to make rapid decisions at the bedside. And we'll do a brief overview of the technology behind how the Accelerate Pheno system, using the Accelerate PhenoTest blood culture kit, which was recently cleared by the FDA for patients in IBD use. We'll talk a little bit about how that works going forward. So let's start here with the urgent clinical need for susceptibility results and getting them faster. If you're anything at all in the world has nothing to do with microbiology, let's pretend for a moment that you are a mom taking her kids to school. Let's pretend that you are a guy working at a tire shop. Let's pretend that you are anybody anywhere in the United States today who's got Twitter, Facebook, and a TV connection. You are hearing about sepsis. You are hearing about multi-drug resistant organism incidents. It's amazing that in the last 2 years, it's gone from something that really was only known in the world of medicine, right, in the world of clinical practice, to where now it's something that's on NPR, and it's something that's on your local news stations, and there's people tweeting about it who have nothing to do with healthcare. And it's because it is quickly becoming, as the economist Lord O'Reilly in the UK for the National Health Service commented, it's becoming the leading health issue of the next 100 years. It is considered to be, by the CDC, by the World Health Organization, and even by the UN, to be the critical issue facing the global economy. And this is because not only the things that we know today about the mortality rates of sepsis, how it gets much worse when you have multi-drug resistant organisms involved in the infection, but it's because of the economic considerations of what happens in a world if we don't have antibiotics to use. And the thought that we could possibly be back in the dark ages without having medicine to treat the common infections that we have. But if hearing it on the news and if hearing it from global well-known bodies such as governmental organizations like the UN isn't enough, here in the US, we've got the stick. We've got the government telling us that you either demonstrate a robust system for stewardship, that you demonstrate how with stewardship you're going to properly and responsibly use antibiotics, or we will pull you out of reimbursement from Medicare. Now this is a proposed rule. We don't know what the final regulations will look like, right? Particularly in today's political climate. But this is not very much carrot and stick. This is pretty much all stick, right? And so where we are here in the U.S., knowing that reimbursement is so heavily tied to everything we do coming from Medicare, this is a significant issue for us all. So each of you who are here have some role in antimicrobial stewardship, right? You are either here as a trained ID pharmacist, as we have Ashley here in the back. We have infection prevention directors here, such as Jared. We have people here who come in as medical doctors. factors. But if you're working in the lab as a microbiology manager, as a med tech, and you don't formally have a position on a stewardship committee, I submit to you, you are critical to the role of stewardship. Because every decision, every piece of data that you provide to the laboratory and the time in which you provide it makes an ultimate impact on how stewardship decisions are made, on that larger picture of the strategies taken and implemented to treat critically ill patients. And where we come in is to provide that diagnostic information more rapidly, particularly with respect to the susceptibility test and the MIC. And ultimately to get those pieces of information faster so that your clinicians can make evidence-based decisions using clinical decision support software tools or other engineering controls that you may have, such as redlining individual antibiotics, so that you can make those informed therapy decisions faster to treat the patient, improve outcomes, improve quality of life for those who are able to get an extra 3, 4, even 6 months out of being able to be treated correctly the first time as quickly as possible. And so ultimately this is looking at improving health outcomes, but it's also looking at reducing costs. Because we don't live in a world, an ideal place that's free of the constraints of costs and free of the constraints of money. And treating patients faster who have bacteremia, treating patients more rapidly, being able to identify, but more importantly, get the right antibiotic at the right dose, with the right dosing strategy, with the least collateral damage to the patient, it makes economic sense as well. And we'll look at some papers that speak to the implications for ICU bed costs and for the cost of antibiotics as well. So let's look at some of the science behind diagnostics and the role of clinical therapy and how they interplay with each other. The first is, let's look at papers that talk about reducing mortality in patients with bloodstream infections. I think this is a requirement. If you ever sit down in a room to talk about bloodstream infections, you see much enough, right? Very, very plainly put, so that familiarity doesn't breed contempt here. Every hour that effective therapy is delayed for the patient with a bloodstream infection, back to Dr. Kumar's paper, showed a 7.6% decrease in survival rate every hour. And so that means the clock is ticking, so that if we can save 2 hours, 4 hours, 6 hours, in our case, 40 to 50 hours, you are getting on the right side of that curve to impact that patient's outcome, to impact the difference between life and death for the critically ill patient. Every hour counts for the patient with bacteremia. Looking at length of stay. This is a graph that's been, a table that's been adapted from Dr. Angela Wong and her colleagues' paper from 2013 at the University of Michigan. And what you're looking at is a quick numerical representation of how they implemented rapid ID technology using MALDI, the traditional MALDI method from isolate, together with antimicrobial stewardship. And this is an important thing. No diagnostic tool is an island. It has to be implemented together with the people who make those decisions on how results are taken and given to the intensivist who's treating at the bedside. So her paper doesn't tease apart those differences. Both things are being looked at. Rapid ID technology, how that improves susceptibility testing results a little bit, but also generally how is it used in the context of stewardship. And so what we see here is that they had a significant decrease, looking at the top line here, in time to result. But more importantly, using antimicrobial stewardship, they were able to decrease significantly ICU length of stay, which is bolded, and significantly decrease 30-day mortality. And there was an association, although not a true correlation, with total overall hospital length of stay. But you notice those are very marked decreases, saving 6.6 days in the ICU for that cohort of patients with bacteremia. Now, how did they do that? It wasn't just the time to ID. These bottom lines are what's important for you if you are sitting here today as an ID physician, an ID pharmacist, an infection preventionist. It's when did therapy actually get changed? When were you able to escalate to effective therapy for that smaller cohort of patients that are on empiric therapy, but none of those antibiotic agents are covering the infection or are active against the etiological agent involved? And then for that broad cohort, really nearly every patient who's being treated with empiric therapy, how quickly can you get them on optimal therapy, to de-escalate, to discontinue unnecessary therapy, to get them on the right drug, at the right dose, with the least collateral damage to the patient. And the key thing is it has to be a partnership between the laboratory and the stewardship team on how those results are going to be used repeatedly, and how are they going to be used so that decisions can be made days, nights, and weekends for the critically ill patient. And this is another piece of data from her paper. When they look at what parts of diagnostic information were used to change therapy decisions, The MIC value, together with susceptibility, was required more than half the time to be able to narrow therapy. The gram stain and identification have great value for escalating to effective therapy. That tends to be a pretty small cohort because most of your institutions, 75 to 80% of your patients are on effective therapy. Those are numbers that have been established through 4 different papers that we reference, whether it's the long paper, Catherine Perez at the Methodist Health System in Texas, Redamar from University of Sita in Spain, as well as Zhang 2015, also from Michigan. So each of these have shown broad agreement that about 75 to 80% of your patients are on effective therapy. The key is, though, how can you de-escalate? How can you do the things that prevent the downstream effects, such as C. diff, such as additional cases of multi-drug resistant organisms that happen because you are selectively creating the pressure to induce antibiotic resistance? So how do we ultimately take this together in advanced stewardship? Again, let's talk about C. diff because the government's talking about C. diff, because your administrators are talking about C. diff. You're probably, in fact, sick of hearing the word C. diff, right? You're hearing this all the time in your stewardship meetings. I like to talk about this because this is a fantastic paper. This is, as a big numbers geek, this is a retrospective study that was done at Kaiser. This was looking at the entire Kaiser network. This was published just at the end of 2015 in December by Tartoff and colleagues. Retrospective review of 403,000 plus patient records. Who wanted to be the biostatistician for that project? Right, that's some work. And there's a lot of great insights here where they tease apart different correlations and associations between what's happening with the patient and when does C. diff incidence increase. This is one of the key findings. Looking at the graph on your right, you see the rate that the incidence as a percentage of the entire inpatient population of C. diff and the simultaneous number of drug classes administered. And when you go from one drug, which is the red column, to 3 or more drugs, which is the yellow column, it's a 5-fold incidence in C. diff with a very strong statistical correlation. There's many other insights that I won't take the time to go in here, but just go back, look at the paper, and see some of the additional insights they have on specific antibiotics such as fluoroquinolone use and vancomycin use and the increased risk of C. diff. It impacts patient outcomes. It impacts the spread of C. diff for every grandmother and every daughter and every niece and nephew who's walking in and out of that room. It impacts the hospital-acquired spread of C. diff for every healthcare worker who's going in and out of that room. But again, it has impacts for you as stewards on reducing antibiotic usage and costs. If you can get those patients on the right therapy and de-escalate faster, that patient can be moved to your step-down unit. That patient can be de-escalated in such a way that they are less of a risk of getting C. diff. Another great paper we're not going to look at, but look at this recent paper that was published in January of this year from the UK National Health Service. A great retrospective review that followed the patients through 2 or 3 different regions within the NHS and had very strong correlation to reducing penicillin use and fluoroquinolone use specifically, and how that tied back to lower C. diff rates in the UK. But again, it all comes back to the bottom line too, right? Because you have the Healthcare Acquired Condition Reduction Program, the HAC Reduction Program, that is now measuring C. diff as one of those 8 core measures under the PSI 90. And if you are not in if you're in that bottom 25%, I should say, you're being faced with a 1% cut in all Medicare reimbursement. So again, not to keep wielding the heavy stick, I'm just the messenger, don't shoot the messenger, right? This is important. And part of what we do is partner with you not only to escalate to effective therapy, which is critical for the mortality benefit to patients, but it's also to reduce these downstream effects for patients who can get off of all of those additional antibiotics as quickly as possible. This is an example of some of the results that we have from our system. This is a patient record from our clinical trial. And this is the kind of patient that when we talk to a couple of our ID physicians and ID pharmacists that we have on our scientific advisory board, Jason Pogue out of Detroit Medical Center, Jim Lewis out of the Oregon Health Sciences University. And when they look at this, they say, this is the kind of patient we see every day. This patient comes in, at least 2 of 4 SIRS symptoms show, they draw lactate, they draw 2 sets of blood culture bottles, and most likely they're moved into the ICU if they're not already there. But the results come back and you have something like this. Looking on the right-hand side, you're seeing that everything here is susceptible based off the MIC for all of the different formulary choices that we have available on our system. And if you have access to an RUO agreement, you would see colistin results here as well, because we also test for colistin. When you have pan-susceptible results, I'd like to, I know we have an ID pharmacist in the room, so Ashley, if you don't mind, I'd like to put you on the spot here. You get this patient back, how quickly from the lab today, how quickly would you get results back that have the MIC with susceptibility normally?
It could be anywhere between probably on when they received it. You could maybe at fastest get it 48 hours.
Okay.
Maybe 30 hours if it right away.
Okay.
I don't think it would be sooner than that.
Depends how fast it grows.
Yeah.
Yeah.
Just how
Yeah.
fast, and then also what time of day they receive for a specimen.
And so Ashley, you're waiting, let's just call it 48 hours. That tends to be pretty quick. That's even what Mayo showed in their recent study. They had 48 hours of susceptibility result. You get this result, you have an E. coli that's pan-susceptible, the patient's stabilized, not seeing any additional problems. What would you do for a treatment decision for this patient?
Well, I look at the antibiotics and see what they're on, and if possible, I de-escalate.
Okay.
Because a lot of our patients will go on broad spectrum to begin with, and then both gram-negative and gram-positive, and so we want to take things off at that point.
Is it possible you might have had a patient like this on an anti-pseudomonal as part of her hair therapy?
Oh yeah.
So you might be able to remove at least 2 antibiotics. They may even be on an antifungal, although, what is that, 5% of the time?
Yeah, we're very low. We don't have, we have more traumas and orthos, less transplant, and like oncology. That'd be more of that population for that antifungal one.
Okay. Well, good. I'm glad to hear that. I like to always ask the ID pharmacist in the room first, and then I put up what they said too. Let's see if our other ID pharmacists agree. It's the exact same thing, right? The first insight from Drs. Pogue and Lewis is, if I know that MIC and I see where I'm susceptible, it doesn't just give me 1 or 2 options. I can look at all the different choices based off of what I know about this patient. Because again, it's not just the susceptibility results, it's that whole host of factors that you have available in the chart. What are the other comorbidities? What else is involved? But knowing this, I can de-escalate maybe to a 1st or 2nd generation cephalosporin. I can put them on something very, very low bioburden to the patient that is going to reduce the risk of C. diff for this patient, as well as get them back down and step down quicker, perhaps even on oral antibiotics, right? So these are the sort of insights you can have with our system, again, including being able to get colistin results where needed for a multi-drug resistant organism. Here's another way to think about this. So I didn't fix my math, forgive me. Let's assume you have 4 patient bacteremic patients per day, because that's the only way the numbers work. The graph on the left shows that this is a month of patients where you're assuming 4 unique positive blood cultures per day, so 120 in a month at your institution. And this is an average for U.S. institutions. Many of you come from much bigger institutions. Many of you come from institutions that are sort of right in this spot. Based off of the long paper, again, 52% of the time, you would need to have the susceptibility result to be able to escalate. So if you're getting those faster AST results, again, based off of our clinical trial data, 40 hours faster than current methodologies across a who's who of clinical institutions, and if your antimicrobial stewardship team is ready to act, has the tools in place, has the resources in place to be able to act across all shifts, can you begin to accelerate therapy decisions faster? Can you maybe move from only changing therapy when your pharmacist makes rounds in the ICU every day to begin doing it within every couple of hours, right? This is the kind of thing that you can do with this tool. Again, rapid ID helps. We don't claim to be a fix-all. If you have molecular technology or MALDI, then you're getting a significant benefit because the ID can help to de-escalate. But how many physicians are de-escalating on ID alone? How many will accept that data and say, yep, okay, I'll de-escalate? The AST is what's needed. So then finally, reducing the cost of care. The ICU length of stay is the largest cost driver for all of your institutions when looking at treating a bacterial patient. It's a fact. It's the nature of the cost of care today in the American hospital. One day in the ICU ranges from $2,500 for a rural institution to $15,000 plus for a specialty children's institution or oncology institution. And so this is up to 60 times the cost of a single PhenoTest blood culture kit. The economics end up working out well because what we're looking at is not simply using this for a very small percentage of patients, but this is able to be used for 90 to 95% of your patients based off of the prevalence of the organisms that we have on our panel. And this helps you to reduce not only ICU costs, but again, on a much smaller level, antibiotic costs as well by de-escalating and discontinuing unnecessary therapy. So your net hospital savings becomes what you save in the ICU, plus antibiotic costs subtracted from the net lab costs of bringing in the Accelerate Pheno system. So here's how the system works. Just as a quick overview, it's meant to be fully automated, be able to run very simply and easily by any technician in your lab. It's MIC-based direct susceptibility results direct from the positive blood culture specimen to help you to expedite antibiotic therapy and personalize optimizing therapy for each patient based off the MIC. And the scalable design allows us to put this in very small institutions to institutions who are running 8, 10, 12, 20 positive blood cultures per day by being able to have multiple units interface together. You get results in under 90 minutes for ID, and then just under 5 hours later, you're going to get the MIC susceptibility result for a total time to result of under 7 hours. Our clinical trial average was 6 hours and 41 minutes. And within the 95th percentile of all results reported, we were under 7 hours and 5 minutes. So we say results in about 7 hours. You'll never have any result take longer than 8. It's that fast, and it's that reliably fast and reproducibly fast for your clinicians. Takes 500 microliters of positive blood culture specimen, transferred to the sample vial. The user pushes everything into the system and presses start. No additional steps, centrifugation, pipetting, washing with a lyse buffer. It is that simple. And the reason is we want to be able to empower many of you who have these centralized labs to be able to decentralize when possible and bring testing closer to the patient at the patient bedside. This can be anywhere that there's a blood incubator, right? Any place where you've got a blood culture system like a backpack that you could bring in the Pheno system and run it as well. We have, in addition to the ID and the MIC result susceptibility, we provide this monomicrobial alert which rules out the polymicrobial infection. It's a powerful additional piece of information that no one else provides. When our system reports an ID target and says this monomicrobial result, it means that you can rule out with 99.6% positive predictive value the presence of a polymicrobial infection. This means you can de-escalate now. It's the trigger to de-escalate immediately when you receive this result. And for most of you running this system, 80% of the time plus, you're going to get a monomicrobial call because polymicrobial infections typically make up 5 to 10% of all infections with patients with bacteremia. Now, of course, in the respiratory world, with lower respiratory infections, polymicrobials are what rule. We are, with our polymicrobial capability, we report up to 3 ID targets, and then we will report the dominant AST for a mixed infection. We'll just report with the 1 AST with an MIC for that dominant organism. We have future work already being done, and we're working on putting together the CE submission in Europe for lower respiratory, for BAL and ETA specimens, and we're continuing to do the initial research work on skin and soft tissue as well as surgical site infections. So this is a system that will grow and continue to expand for all of the critically ill patients who are suffering from bacterial infection. And again, we also do cover a few targets for fungi as well, for looking at candidemia. Another way of just seeing how the poly works, we use a mixture of universal probes and then specialized FISH probes that target based off of a conserved 16S rRNA sequence. And what we're doing is being able to then light up and co-locate signal based off of which signals we get that light up as bacteria, and then the specialized target probes that light up based off of an infection. So here we're looking at an example on the right of a mixed infection of E. coli with Citrobacter. We can clearly and easily identify which organisms are which, and then reliably provide the ASC for the dominant organism as well. If you haven't seen this before from one of our commercial development directors, I think this one screen is the best and quickest way so I can give time for all of our speakers today to say what it is we do. We're taking thousands of time-lapse, high-definition images of each individual cell that's diluted and introduced into the flow cells on our consumable system with our PhenoTest blood culture kit. And that's what you see in the lower right corner. We then generate 30 different morphokinetic feature analyses. These are everything from size and shape of cells as they grow, the size and shape of the entire microcolony, how the daughter cells divide, but that's the morphology piece. On the kinetic side, we measure cytality, we measure division rate over time, we measure kill rate over time. We're looking at all of these different pieces of information so that we generate the information that ultimately correlates to a very broad library of tens of thousands of known templates of MIC response. So that from a single antibiotic concentration, we can provide that reliable MIC that then is translated into the customizable MIC interpretation whether you're using CLSI or some other set of interpretation results. And this is the key input for PK/PD calculations for where it's needed, right, in the lab, particularly for your ID pharmacist who needs this to calculate based off of therapy. And what does this ultimately do then for you? It's reducing the time from empiric to optimal therapy. This graph represents not an Accelerate promise, this represents the comparison at 13 different clinical sites, looking at over 1,800 specimens that were run, and we were over 40 hours faster on average across these 13 sites that are really a who's who of clinical practice. So when you're here today, you're listening to Krista Pardue, to Dr. Walk, who have implemented this technology and are beginning to measure the results, who are beginning to see how the results actually impacted specific patient lives, giving you the background and data. But I encourage you, ask them questions as we have time at breaks today and hear from them about how they're using the system in their lab. And again, we have a host of papers and references that we can provide for you here at the conclusion of our day today.
Thank you, Levi Kirwin, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type your question into the box that appears on your screen and click on the send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, we use the Antibiogram at our facility in conjunction with ID. Why would I need ASD results to help the doctor choose therapy?
Okay, thank you. Thank you for that question. Yeah, so good question. This is a very common thing, right? The Antibiogram has been relied upon as the best sort of rubric, right, or the best decision-making tool for helping physicians to choose an antibiotic course or antibiotic regimen in the absence of complete microbiology diagnostic results from the lab. And the reason why they've chosen this, right, is because when you think about traditionally how a physician would receive results from the lab, they don't get everything up front. You go back 50 years all the way through to today, and unless they have an Accelerate Pheno system, then what they're doing is they're waiting 12 to 24 hours to get the first acknowledgement that the blood culture bottle has popped positive. So the patient is already on empiric therapy because they've matched 2 of the 4 symptoms for SIRS. Their condition has not improved, and so they've been started on 2, 3 antibiotics for gram-positive, gram-negative coverage. And in the case of certain high-risk patients, they'll have additional gram-negative coverage for anti-pseudomonal, like, for example, cefepime. And then they'll be added to that for, again, a smaller category of high-risk patients. They'll be on an antifungal of some kind, like amphotericin B, for example. So they're waiting for results, and they get them piecemeal. They get them spread apart over a significant period of time. The blood culture bottle result is between 12 to 24 hours before they get that result called into them. It's another 1 to 2 hours before they get the gram stain result called in. And it's a critical result. The standard's 1 hour, but it can be longer. And then they're waiting for ID results. And this varies widely from laboratories that have recently adopted a molecular platform. They might get the result called in 2 to 4 hours. MALDI, it's going to be 24 to 30 hours from waiting for the culture. And then for all of the traditional microbiology systems for ID, they're waiting between 24 to 36 hours between the time that the solid media has enough organisms on it to show growth, and then they test. And this is still waiting another 24 hours before they can get susceptibility results, right, whether they drop discs or whether they're going on to a traditional susceptibility platform like the VITEC or the microscope. So the challenge is, what do doctors do when they don't have perfect information? Well, they develop the Antibiogram to look at the historical record of the susceptibility profile of organisms that are most commonly involved in bloodstream infections. And you look at the challenges of the Antibiogram. All the Antibiogram does is it tells you what historically, going back a year or 2 years, by specimen source in some cases, otherwise it can be all lumped in, what is the most common susceptibility of an organism by antibiotic that the hospital chooses in the formulary. And when you're looking particularly at gram-negatives, you're looking at organisms that typically are only 70 to 75% susceptible to the common empiric therapy agents. There's been several research papers out of the University of Michigan, Methodist Health Center, and other places that have shown that empiric therapy only is effective therapy about 75% of the time. So what they're doing is making a best guess. But that means 25 to 30% of the time the patient is not on effective therapy. All of those antibiotics are not working. But by using the antibiogram, they're doing the best possible guess, right, using the science that they had available to track historical trends. The definitive result, though, for getting the patient on the right drug at the right dose with the least collateral damage based off of adverse effects is providing the MIC result with a susceptibility interpretation based off whichever guidelines you're using. So the antibiogram is good when you don't have a faster phenotypic susceptibility test to tell you what to do. But now laboratories that are using the PhenoTest system, they're getting all of those results together at once, getting rid of the delay between when the tests are posted into the patient's electronic record and when the physician can act. And so this means that getting the AST result together with the ID in less than 7 hours from blood culture positivity, the physician can make a decision based off the evidence for that patient, customized by individual choice based off of that patient's profile and that patient's biology, rather than using a historical record. And so it's just where science is now leading medicine to go in terms of best possible practice with individualized therapy and individualized medicine. And so again, that's where the PhenoTest can advance the practice of medical care in the hospital. Thank you.
Thank you. Our next question is, we have a BioFire system that gives ID and resistance markers in about 2 hours. Does your system provide evidence of resistance?
Okay, great question. So the identification results that the BioFire or that the PhenoTest blood culture kit would give on the Pheno system, you'll still get a complete panel of gram-positive and gram-negative targets. We also add the 2 most common Candida species targets that are involved in candidemia. But the resistance marker, all a resistance marker does is give you a positive or a negative call for a resistance gene. We go further than that by providing the complete susceptibility profile by organism and antibiotic combination with the MIC together with the susceptibility interpretation based off whichever susceptibility guidelines you're using, which for most U.S. labs will be the CLSI guidelines. For international labs, this may be the UCAST guidelines. And so what this means is, think of it this way, the resistance marker, right, and particularly with gram-negatives, if it's a positive result, it tells you that the resistance marker is present that rules out a certain antibiotic or a certain group of antibiotics. It can rule that out, but when it comes to particularly gram-negatives, a negative resistance marker result does not rule in the capability to provide and to go ahead and dose with a more narrow spectrum antibiotic. And here's the reason why. There are porin mutations, there are efflux pumps, there are different conformational changes in the membrane that are observed, these known mechanisms that are outside of genotype that are phenotypically expressed when organisms, particularly in the gram-negative Enterobacteriaceae class, are exposed to antibiotics. And so what phenotypic susceptibility does is get beyond those limitations. And further, rather than giving a presence absence, it gives the MIC, which is that definitive diagnostic result for choosing the dose, the dosing strategy for the given antibiotic, and at the least collateral damage possible. So this allows a clinician to see at a glance what the organism is susceptible to so they can choose narrow spectrum antibiotics. They can de-escalate, whereas the resistance marker choice really can only be used for escalation in a very large number of patient cases given additional comorbidities. So the susceptibility result being the definitive diagnostic result for choosing an antibiotic, having that MIC available allows the clinician to then de-escalate and reduce the impact of selective pressures that lead to multi-drug resistance in that patient, being a locus for the spread of multi-drug resistance elsewhere in the hospital. And most critically, given today's healthcare environment, it reduces the pressures that produce C. diff infections. And there's significant numbers of papers that show correlation between multiple classes of antibiotic and C. diff infection. So having that result, it does more than just select the right antibiotic for the patient today. It means we have antibiotics for patients for the future as well. And so that's where we extend and go further than what just a resistance marker alone would do. All right, thank you for your question.
Thank you. Our next question is, we've implemented MALDI, a new VITEC 2 and Veregene in the past 3 years. Each time it took nearly a year to complete verification and IQCP. How long does it take to complete verification with the Pheno?
Okay, great question. You sound like a microbiologist who's actually lived this before. I'm guessing you may have lost a little hair from pulling it out as you were working to get one of these systems verified. Listen, MALDI and some of the molecular tests have revolutionized microbiology in the past. These technologies that are now between 5 to 15 years old have meant that you can get a faster ID result. But when they were introduced into the laboratory, there really was not a lot of thoughtfulness on how to convert a life science research technology into something that a clinical microbiologist could quickly and rapidly assimilate, teach to a med tech, and then have them begin to use. And so the verification protocol, as I'm sure you know more even than I do, is the verification protocol requires you to test across the set of targets, in this case ID targets, that are tested on the system, as well as in the case of a molecular platform like a BioFire or a Veregene, you also have to test a variety of resistant and susceptible organisms for those resistance marker targets. What we've done at Accelerate is we've looked back at the historical record on how verifications have been done by interviewing hundreds of microbiologists around the world, and then getting what are those pain points and what are those challenges that are needed to make a verification go faster. What has slowed down a verification in the past, and how can we help? So what we did is we put together a complete performance verification protocol that has a series of different choices that are pre-mapped out that include the right seeded isolates to test across the breadth of resistance or susceptibility by organism, as well as to thoughtfully select the right organisms across a panel to make up for gaps in your local etiology, where, for example, you may not see Acinetobacter cases, right, or Serratia cases more than once or twice in a year. And we've created that so we can provide all the organisms up front. We provide the support with a trained microbiologist who we have hired as a technical application specialist. And so what we've done is we've created a protocol, and in fact 3 different choices of protocols that can be run within 60 days. When you take into account additional data analysis and capture and then final decisions, typically it's 90 days to go live from the time that verification starts. And so we found that looking at how microbiology has progressed and advanced while taking into account the recent changes in CLSI standards and the IQCP standard from CLIA as of 2016. We're taking into that account so you have a single verification that supports IQCP and supports go live within 90 days. And if you have any questions about that or want to see examples, I have my contact information here, and I'd be happy to chat with you. Thank you.
Thank you for that answer. It looks like we have time for one more question. Our last question is, how many places in the U.S. are using the Pheno system?
Okay, great. So right now we have over 200 modules placed worldwide. We have in the United States, we have nearly 180 modules that have been placed or in the process of being installed. Of those, we have a select handful that have completed verification and are they actually have gone live and are reporting patient results. But this represents over 40 hospitals in the United States that are currently verifying or have gone live with the Accelerate Pheno system. All right, thank you for your question. I think we have time for one more question, looks like by my clock. Let's go ahead and try to get that.
Okay. Our last question is, How much time does it take to run each test? How many tests can you run at once?
Okay, great. So a test is, we provide the identification result, and that crosses to the LIS within an average of an hour and 21 minutes, or think of it another way, about 1.3 hours. And then the system automatically continues on to AST results, and that AST result's provided on average about 5 hours later. So the total time to result, you get the ID and the susceptibility result based off of our clinical trial. The average was 6 hours and 43 minutes to time to result. So again, ID comes first, the susceptibility test result comes later. The system is designed to have up to 8 modules connect together and pass through a single LIS interface. So a lab can run up to 8 specimens at a time from a positive blood culture. And again, as a reminder, the positive blood culture, it's 2 minutes hands-on time maximum between loading the sample vial, loading the PhenoTest blood culture kit into the system, and pressing start. And I encourage you to go to our website here after this, or here on the or use the virtual demo that you have here on the LabRoots site, and take a look at how that works and how the loading of the system works if you have any more questions. Okay, thank you.
I would like to once again thank Levi Kirwin for his presentation. I would also like to thank LabRoots and Accelerate Diagnostics for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now, and thank you for joining us, and we hope to see you again soon. Goodbye.